Today we're going to illustrate how to use mixture models for clustering using a real data set. That real data set is called the IRIS data set. It's a very famous one. It was collected in the mid-1930s by a biologist whose last name was Anderson, and then it was analyzed by Fisher in a famous paper in statistics. So this data set contains 150 observations, 50 for each one of 3 species of the iris plant, and we're looking at the flower of the plant in particular. For each flower, we have 4 different characteristics: the length and the width of the sepal, and the length and the width of the petal. And the 3 species of the iris plant that we're looking at here are Iris setosa, Iris versicolor, and Iris virginica. If you just do help of iris in R, you can see the information that is here on the right-hand side of the screen. So this is another data set that is included with R. Okay, so let's start by just plotting the data to get a sense of how this data set looks like. So we have these 4 variables per observation. So what I'm doing here is a scatter plot where I looked at every pair of variables and a scatter plot of the 150 observations for each one of them. And I'm using both colors and letters to represent the true cluster membership. So in this case, we know what the species the plants belong to beforehand, and we will use that to test the results of our algorithm to try to understand what the algorithm is doing, or different algorithms are doing. And a couple of observations are probably important here. So the first one is that you can see pretty much in every scatter plot that the setosa flowers are clearly different from the other 2 species. So they definitely have a very different petal. They are completely apart on both characteristics, and the sepal is also slightly different, at least the sepal width. The other thing that is worthwhile noting is that even though the other 2 species are different, there is some overlap between the plants. And in particular, you can see that if you were to look mostly at sepal length, the distinction between, or just look at sepal length and width, you would see that versicolor and virginica are really, really hard to differentiate based on the sepal. Really, the difference comes from including information about the petal is really what allows you to distinguish these 2 species of plants. Okay, so this is the truth that we will eventually be comparing against. Now, what we're going to do is we're going to fit or use 2 different approaches for this problem. First, we're going to use a mixture of normals. In this case, we're going to assume that we have 3 components in the mixture, and we're going to fit a mixture of 4 varied, tetravariate, if you will, Gaussian distributions that allow for different means and different variances. And you can see how allowing for different means and different variances is going to be important here. So the variance of the for example, virginica and versicolor, you can see that they are very elongated and they are clearly kind of diagonal oriented in this direction. Whereas if you look at setosa tends to be much more, the variance-covariance matrix tends to be in some sense much more compact, as you can see in these 2 graphs. So it's clear that the different components of the mixture, the different clusters, will have very different shapes. And so a more flexible model like a mixture of Gaussians that allows for mixing in the variance-covariance matrix is going to be important. We're going to try to compare the results from that mixture model with those that you obtain by using k-means clustering, which does not allow that different variance-covariance matrix, and actually doesn't even allow the different dimensions to have different variances. So this is a nice test case in which we can show that allowing for this more general model actually leads to better clustering results. Okay, so otherwise the code that we're going to use is very similar to the one we have used in the past, so you will be very familiar with this. We're going to do 15 runs of the algorithm just to make sure that we actually reach a global mode in the algorithm. So let me do that. Sorry, we need to provide all the information to the code. So now that it is running, it will take a few seconds to finish. And so it's currently on the first try. Now it started the second try. In every case, the initial point of the algorithm is different. And so in some cases, it's faster and it converges in fewer steps because the initial step or the initial iteration is closer to the mode of the algorithm. So let's give it a few seconds for it to finish running all the different iterations. What we're going to do once it's done is we're going to pick the best iteration, the one that leads to the highest value of the Q function, and we're going to try to use the values of the probabilities of membership to each one of the clusters to decide on the indicator C. Okay, so now that it has finished, I'm going to decide for the best run out of the 15 what is the membership of the observations. And then the next thing that I'm going to do is I'm going to plot the observations. Okay, and so now what we have is essentially something that looks a lot like the previous graph where we're trying, but the numbers now correspond to just the cluster that the algorithm identified. And we can see that, again, it has separated what was setosa. So if you remember, these clusters down here are setosa. So those have been separated reasonably well from the rest of the observations, so no problem there. And it has also been able to separate Virginica and versicolor reasonably well, but we need to still investigate in a little bit more detail what is happening down here. One way to do that is by using the adjusted rand index. This is a measure of how well or how close to each other two partitions of the data are. And what I'm doing here is I'm comparing the partition that we generated against the true partition of the data that is given by the species indicators. And if you remember what the adjusted rand index is and how to interpret it, the adjusted rand index can take values of up to 1. And if it's 1, it's because the two clusters are identical or the two partitions are identical. And then values that are close to 0 indicate that the two partitions are not really looking similar. Okay, so if we compute the ARI, we can see that the ARI in this case it's 0.9, which is a reasonably large number. So we're not perfect. We're not matching exactly the true species classification, but we are pretty close to the true one. Now I want to contrast that against the results of just doing k-means clustering. And there is a function in R that allows you to do k-means clustering, just k-means. And one of the things that you need to give to this function is, again, the number of clusters. We're also going to use 3 for that algorithm. And we need to give it how many repetitions of the algorithm. In this case, we're going to use 15, just like for our EM example. And again, we're going to run this and compute the adjusted rand index. So again, we run the algorithm, we pick the best, or what this function internally does is it picks the best of the 15 runs. And again, we display the data. Not surprisingly, it's not hard to separate setosa from the other two. But we can see now that the separation of virginica and versicolor, it's a little bit odd. And we can verify that the results are not as good as the ones that we had obtained with the mixture model by looking at the ARI. And you can see that the ARI in this case is only 0.73, which is much lower than this 0.9 that we had obtained before. In other words, the partition structure that comes from using the k-means algorithm looks less like the true partition than the one that you get from using the more flexible mixture model. So this is an important observation. If your data doesn't really have the characteristics that are supported by the k-means clustering algorithm, then you're typically much better off by trying to use a more general algorithm that is based on mixtures.